Good afternoon, everyone. My name is Joel Tickner. I'm a faculty member at the University of Massachusetts Lowell, and I direct the Green Chemistry and Commerce Council. I'd like to welcome you today to this afternoon's webinar of the Green Chemistry and Commerce Council's Green Chemistry Education webinar series entitled Integrating Toxicity Information into Chemical Design. For today's webinar, I'd like to introduce Saskia Van Bergen, who directs our Green Chemistry and Commerce Council's Green Chemistry Education Group. Saskia is the Green Chemistry Coordinator for the Washington State Department of Ecology. Saskia, welcome to the webinar today.
We have three great speakers who will be providing traditional perspectives of a chemist, strategies for improved design, and examples of molecular design guidelines and how they can be used for safer chemicals. The three speakers are Marty Mulvihill, who is the Executive Director of the Berkeley Center for Green Chemistry, Jakub, who is the Chief Scientific Officer at Sustainability Agency, and Nigel Greene, who's an Associate Research Fellow in the Compound Safety Prediction Group at Pfizer. Let me introduce Marty Boyle, who will delve into the mind of a chemist and provide strategies for improved design. Marty?
It's a pleasure to be here today, and it's a pleasure to be talking about a topic that I've spent a lot of time thinking about in my role here at the Berkeley Center for Green Chemistry. As the executive director, one of the things that I do, and that one of my favorite parts of the job, is teaching graduate students, both in chemistry and also in other disciplines, how to think about the design and selection of safer materials. Along those lines, the two questions that motivate me and that I hope to address in today's talk are, first, what do we need to do to provide the next generation of chemists, the folks actually making the molecules, with the information that they need to develop a robust intuition about what may and may not be hazardous? And the second part is, how do we promote the adoption and the commercial success of these safer chemicals during the design process? There's chemistry involved in that too, but I'll actually talk about how in the first case, my goal is to really develop an intuition, a gut feeling in my chemistry students about what makes better and maybe less good chemical design. Whereas the second is really all about teaching people, chemists, engineers, public health folks, how to search the data and how to think about both technical performance as well as health and environmental performance alongside one another as you design a new chemical or product. So to start, I'd like to share something that I found just a couple weeks ago. So this is literally an excerpt from a page, an exchange that happened on DoE's Ask a Scientist webpage. And I think there's a lot that we can learn from the way a chemist addresses this problem. So there was a teacher from North Carolina who posted to the website asking for a substitute for toluene in a high school laboratory. And that's really how she asked the question, what can I use to substitute for toluene in a lab about polarity? The four answers she got are exactly the answers I would expect if I asked most of my undergraduate chemists that same question. The answers really are related to, one, where do I find this stuff? Why are you substituting toluene in the first place? Because toluene clearly already worked in the lab. So can I help you find it somewhere else? Two, just using simple chemical intuition about polarity of toluene, suggesting either my favorite answer, so 2 and 3 toss off a particular decent substitute for toluene if what you're thinking about is solubility or polarity. And I think you see that explained most clearly in answer number 4, which says, okay, here are the solubility indexes of each of these molecules. Now, hazard doesn't come up anywhere there. But one thing I've learned talking to high school teachers is that's often one of their first concerns when they're thinking about how they're going to actually teach chemistry labs in a public school setting, or really in any K through 12 setting. One of the biggest concerns actually has to do with how they deal with the waste, if it's safe to come in contact with their students. And you'll notice that doesn't come up here at all. And I'm not surprised because it wasn't clearly articulated in the design of the question. And that's something we'll come back to later. But for now, I want to explain how, just in a couple brief slides, we got to this point where if you ask a chemist for a substitute, they're going to give you the most similar in molecular property and or molecular shape substitute that they can think of. It has a lot to do with how we've created and trained our students in modern chemistry. So the point of this slide, and it's a little complicated, I won't stay on it long, but the idea is what we've really been doing over the past 200 years is building up a very strong base of knowledge about molecular level understanding of matter. So chemists, we deal with matter, mostly with molecules, but also, as you can see more recently, we've been excited about slightly larger things like biomolecules and nanoparticles. They're actually much larger than your typical molecule, something that I find is often confused in the public. But what we've done is develop a lot of solid quantitative data and intuition around how things on that scale work. When I walk into a restroom and use a bit of soap, I can tell right away what sort of fragrances and other things are used in that soap. And that's just the way I see the world. I think in molecules, and that's how we teach our chemists to think. At the same time, we were developing this molecular level understanding. The chemistry industry was doing an excellent job of using that understanding to translate what were waste or otherwise cheap bulk materials into things with much higher value. Many of you are probably familiar with the story of BASF, who got its start really turning tar into dyes. That was their first and key breakthrough to become a dye stuff company early in or late in the 19th century. Dow got a similar start, starting with bulk materials, in this case mostly salt, to develop the chloroalkyl process. And although they didn't develop it, they really did an amazing job optimizing it so that it was much more efficient and they could very cheaply produce bleach. Their first patents that all came at the beginning of the 20th century were related to the chloroalkyl process. And it's one of the reasons why we do have a lot of chlorinated and other products in our supply chains, because we've really optimized the ability to turn very cheap things into more complex, more valuable things. And DuPont actually got its start before both of them, doing something very similar, starting with inorganics, in this case potassium nitrate, sulfur, and charcoal, and turning it into gunpowder. And that expertise, again, through that development of understanding at the molecular level, helped these large companies turn very simple, very cheap things into much more valuable, much more high-performing materials. So at the same time we're developing a robust understanding of molecular mechanics and dynamics, electronic structure, molecular structure, we're applying that to real-world problems of taking stuff that's relatively cheap and adding value through simple molecular transformations. This is all to emphasize how we think as chemists. At the same time, and actually even starting probably earlier than what we consider modern chemistry, there's been the development of the study of toxicity, really the study of poisons, and characterizing what materials in what amounts cause what sort of impacts in biology. And so it often started from the observation of the final impact. And in the case of poisons, that's often death. And when you think about what the most common type of toxicity information that are out there, it relates back to this historical development of being worried about poisons, needing to characterize what things in our natural and built environment can lead to negative outcomes. And so it's been going on for a long time. It takes an understanding of molecules, but it really focuses on the biological effects. And for that reason, it hasn't always been written in the same language, even though there's clearly a tie between the molecular understanding of chemistry and the impacts of the molecules that are studied by toxicologists and toxicity until very recently they started kind of at opposite ends of the spectrum. That gets me to what I think is key to bridging this gap and to helping chemistry and toxicology come closer together and helping them inform each other so that we can bridge the gaps between this very deep molecular understanding and all the knowledge that we've built up as chemists and all of the real impacts and all of the knowledge that have been built up on the side of toxicology. So what this little sketch drawn actually by a colleague of mine in public health is our dinner table napkin kind of approach to thinking about if you start on the left-hand side and are looking at the impacts, the epidemiology, why a particular chemical might be bad, there's actually a lot of steps to go through to get back to what you see on the far right-hand side, which looks like roughly a derivative of nonylphenol ethoxylate, to understand how something that causes disease or other impacts in the real world is related to some choice at the molecular level. These are things that are happening. Many people have probably already told you that molecular toxicology and thinking about toxicity at the molecular level is something that has happened over the last 20, 30, 40 years. We've been developing much more information that's specific about various pathways in various species or tissue cultures or getting down to the cell level. So we are beginning to develop a much more integrated picture of what, how molecules relate to the society level concerns. So what I want to do is take that knowledge, take that understanding that has been gained by chemists getting a little bit bigger in their perspective and by toxicologists getting down to the molecular level mechanism and talk about the ways that I try to develop an intuition among chemists. So I basically group my approach into the framework of persistence, bioaccumulation, and toxicity. And the reason why I want to include persistence and bioaccumulation is these are the easiest things to teach to chemists. They are very easy to predict based on molecular level properties and structure. So I actually spend a fair amount of time, and I won't go through it with you all here today, but teaching my undergraduate and graduate chemists how they can take a quick look at a molecule, and while they're characterizing functional groups and reactivity, they can also pretty quickly characterize, is this something that's likely to persist in the environment? Is this something that's likely to degrade? I don't give them, there's not enough at this level to say how long it'll take or what necessarily all of the products will be, but those are things that are relatively easy to predict just using a molecular level understanding. Toxicity, on the other hand, the actual impact within the biological system is harder, and I'll talk a little bit about some of the signposting that I do for my students to have them think about what is it in a molecule that should maybe make me ask my toxicology colleagues for some more information to dig a little deeper. And that's my real hope with toxicity, because although, as Jakub is going to talk about, there are some great ways to tie toxicity to molecular level characteristics, we're still doing that. It's still an emerging field. I can't wait for more of that. Because I think it will be what helps us tie these things back to the chemist at the bench. So I'm not going to talk about this too much, but these are some great resources for helping teach chemists how to think about persistence. Work comes from the EPA. It's a really great way to help undergraduates understand what to look for in a molecule and how well that'll predict its eventual ability to degrade in the environment. Similarly, so if persistence relies a lot on structure, looking for halogens, looking for branching, looking for various atoms and molecular architectures. Similarly, bioaccumulation, or kind of complementarily, bioaccumulation takes a look at maps very well with partitioning. So the ability for a molecule to either go into water or go into oil. So that's represented on the x-axis as log KOW, maps, not perfectly linearly and not perfectly narrow. There's certainly some variation in the data, but it maps very well with the bioaccumulation factor, so BAF, or bioconcentration factor, BCF. So this is, mostly based on data for aquatic species, or in fact this graph is completely based on data from aquatic species. But what it shows you is with a simple parameter that isn't very hard to measure or predict, using your understanding of molecular structure, you can do a pretty good job of guessing how bioaccumulative a molecule may or may not be. Now, this sort of estimation doesn't make up for actually doing the measurements, but it helps develop an intuition, a gut feeling among our chemists about what a molecule might or might not do in the environment. Similarly, there's a lot of good work that has been done translating absorption in the body to simple molecular parameters. So, and I bring this one up because it'll relate to something I talk about a little bit later, but we know that particulates that are less than 5 microns are going to get into the lungs, and in most, if not all cases, they're going to irritate the lungs and lead to adverse outcomes. That's something that we can teach people, teach chemists who understand what that size scale means, exactly what they're going to be looking at, what they're going to be considering. Similarly, if you're dealing with a molecule and you want to know whether or not it's going to be in the air, and if it's in the air, therefore it can get in the lungs, talking about vapor pressure, things that we already teach, can really help students translate into whether or not they might be hazardous as they get into the body. So you can do this not only for the respiratory tract, but for absorption in the gut, for absorption through the skin. I won't take you through all of that today, but it's a nice example of where you can use molecular properties or other molecular level descriptors to help get an insight into potential toxicity. And then finally, when it comes to structure at the molecular level and toxicity, I spend a lot of time thinking about what are the ways that chemicals can react within a cell or within a biological system. And I won't go through these all today, but I do have slides and presentations on all of them thinking about electrophiles, which often can react with the plentiful biological nucleophiles in the body. And there's particular electrophiles that are more reactive than others in the biological system, and that's not always the same as how they react outside the biological system. And there's some fun teaching that we can do about that. Radicals and sources of radicals are known to cause stress in biological systems. Reactive oxygen species are all things that create a radical. So what are the chemicals that lead to these reactive oxygen species? Heavy metals, looking at the fact that how do they act in the body and how can we differentiate between them? And then there are organic cations that disrupt things like cell membranes, and specific chelators or ligands that react with known pathways within the body to cause harm. So there's a fair amount of good work that's been done in all of these areas talking about, at the molecular level, what are the toxicity mechanisms for each of these sort of potential pathways for harm. So it's a way to bridge from harm back down to the molecular level. So the second part, so if that is building up from the bottom up, how do we translate that toxicity information into a molecular level intuition that a chemist can say, hey, look, I have a molecule based on its structure, based on its log KOW, it's likely not to degrade, it's likely to bioaccumulate. And it has some of these other characteristics that may be of concern. What do you do next? That's what I want to do from that first part, and that's really where the chemistry comes in. The second part the chemistry comes in another place. This is really about how do you select safer chemicals from a list of potential chemicals within a product. So we teach this to an interdisciplinary group of folks who are really looking at ways to move this information about risk, hazard, impact much earlier in the design process. And so what we teach here is a much more iterative process where everyone needs to work together. It takes a lot of different types of knowledge to go through this design process. You first have to specify function. And once you specify function, you need to specify it at multiple levels. So that's where you're going from asking that original question in the green box on the left to getting into this cycle where you have to really write out all the requirements, both the molecular level requirements and the ultimate performance requirements. And then where the chemistry comes in is this next little set of arrows where you have to think about investigating what the alternative approaches might be. This is where you have to put on your chemistry hats and say, what are the other molecules that might be able to work within this system? What are the other materials that might be able to work within this system? Is there a way to actually redesign the system so that we don't need any of these molecules? That's what happens there, and again, it takes people with different perspectives to give you the most potential options to then do the next step, which is evaluate the alternatives. And you have to continue going through that loop and checking back in with whoever you're collaborating with, whether you're a company and you're collaborating with the public or someone in your supply chain, but going back, checking in and figuring out, these are all of our options. Some of them are rather incremental, so they're slight reductions in hazard, but they fit into the system very well, whereas others can be quite disruptive and would require a much more significant investment to bring to bear. So this is a very simple example to think about that. If you pose the challenge, how do I get rid of brominated flame retardants within a printed circuit board, you have many, many options. And whether you start down there at the bottom to replace the current PBDEs with a new class of PBDEs, either the tetrabromal bisphenol A, that's TBBA, or the polymerized version of that, you're making some improvements, potentially, on the overall hazard or potential exposure, but you're not fundamentally changing things. These are the low-level improvements that can happen. On the other hand, you can start thinking about completely different classes of chemicals that might be able to, through a different mechanism, retard the flammability of a printed circuit board, of a piece of plastic. Or you could go all the way to the top and throw the chemists out and say, hey, how can we change this so there's not a fire hazard on the printed circuit board? How can we reduce the operating voltage of the current so that we actually don't need to add a lot of flame retardant chemical to this printed circuit board? Or somewhere in between, you could say, hey, let's bring the chemists back on board with the material scientists and have them design a new board that is inherently flame retardant. It would need to be made out of different materials. In order to do this, we take our students basically through a four-step process. The chemistry really comes in identifying the compounds of interest, and then we teach them a more traditional hazard-based approach where we have them screen authoritative lists, we have them do literature reviews, and then we have them fill in the gaps to the best of their knowledge using a lot of the things that I was talking about in that earlier part of the presentation. One of the things that I really want to highlight is that depending on what sort of material you're looking at, there are different factors that influence the overall hazard. If you're looking at plastics, it's not the final plastic you need to be looking at. You need to be looking at the feedstocks, the additives, and the breakdown products. Whereas if you're looking with metals and minerals, the whole P and V kind of go out the door because they don't really change in biological system, but on the other hand, size and oxidation state, what they're compounded with, their form is all critically important to dictating their hazard. And then chemistry and molecules is what I've been talking about a lot, where structural features and partitioning really dictate what's going on. So I'll skip those things because other folks can talk about them. But the last thing I really encourage all of my students to do is move beyond red lists and what's good and bad to thinking about health as a performance characteristic. So this is a derivative of the green screen that we did in our last class that shows how the students find a couple options for each of the chemicals that are needed within any given technical solution, and then characterize them so you can start seeing where your options are, where you can make the biggest impact. And that takes me to my last slide, which is really emphasizing that there are two ways to think about these problems, and both are very important, and they work side by side. There really is the bottom-up approach, where you think about design at the molecular level, and you build that intuition among the people you're working with so that they ask the critical questions when they're evaluating alternatives. And then secondly is really teaching people where in this design process and where do I incorporate hazard data and where do I find that data? How do I search it? How do I find it in its vast array of locations? And that leaves me with a couple take-home lessons, which is never assume that when you're talking to a chemist or an engineer, for that matter, that they're thinking about inherent hazard. Be explicit in asking what you're looking for so that you can help translate the current state of knowledge in a way that really empowers them to take steps forward on a path towards continual improvement. The whole idea for me is that these things are relative. We can take precautionary approaches if we are honest about what we know and what we don't know, but that for any given product or process, we really need to think about what are the next best steps. And we can do that if we marry this hazard-based information approach with a molecular design approach. And I think the next two speakers will dig more into both of those areas.
Great, thank you, Marty. Next, we have Jakub, who will talk about computational approaches to designing safer chemicals.
Well, hello, everybody. Thank you for having me. So when discussing computational approaches to designing safer chemicals, I think the best place for us to start is by defining what we mean by computations and defining what we mean by designing safer chemicals. And for the latter, I think we can turn to Paul Anastas and John Warner and their number 4 principle of green chemistry, which states that chemical products should be designed to preserve efficacy of function while reducing toxicity and other environmental hazards. The important observation I think to make here is that design of chemicals inherently deals with hazards, not exposure. So any models or guidelines that we develop treat exposure as a constant. So I think this is something that we have to keep in mind. It's an assumption in our model. Now, with regards to the statement on this slide, I would make the claim here that at present, the status quo of hazard testing of chemicals is very much different. It's not aligned with the statement. At most, I would say we test commercial chemicals after they have been produced, rather than attempt to design them in a way that would minimize their biological activity. You could ask, well, is it possible to actually, alter a chemical at the design stage to make it less toxic? And I would say yes, I think it is. You can think of computer-aided drug design, and the next speaker will talk about that probably much more. Computer-aided drug design has shown some success at being able to optimize biological activity of a particular chemical against a specific biological target. Now, the idea of safer chemical design, if you think about it, is similar. In some ways, it's the exact opposite. Instead of an active compound, we are looking to design one that is not active. It is fair to say that this is probably much harder to accomplish. Just imagine if a computational chemist can be somewhat successful at optimizing biological activity of one chemical against one target in one species. How can we design guidelines or models, those models that would abolish activity against thousands of targets for thousands of chemicals across many species? It's a big challenge, and we are just at the beginning of the journey. And I think it's a challenge where the 80-20 rule and, in general, probabilistic approach is more relevant than a definitive answer. So, in other words, if to use a metaphor, it's a challenge where a broadsword may be more applicable than a scalpel. And again, it's a fine line we are walking, since chemicals' desired functionality still needs to be preserved in the process. So if the chemical is intended to be a paint thinner, it has to remain a paint thinner. So in principle, we have two types of possible approaches when it comes to dealing with toxicity of chemicals, and both have merits. The value of the reactive approach, which we have been practicing till now, is that we can identify hazardous chemicals from those that already exist. And we can evaluate chemical alternatives, and that is very useful, of course. By comparison, the value of the proactive approach is that we can, in principle, redesign an existing chemical to minimize its biological activity. We can also design a new chemical that has a superior safety profile to other chemicals in the market. Now, while both approaches are very useful and have merit, the importance of the latter one, the proactive approach, I think cannot be overstated. Now, I would say that that's probably the future that we want to be headed towards, because it is easy to avoid a quote-unquote bad chemical, but the irony is that the hazards of high-profile chemicals like BPA, like phthalates, are much better studied and understood than alternatives. So it's important to have better metrics and design tools for new chemicals besides, arguments like I'm not using BPA or I'm not using phthalates. Now, but before getting on with the rest of my spiel, I should make this sort of very clear that the design of safer chemicals is a grand challenge, and the strategies I will outline in my talk are just a starting point, really. So they do not yet specify how you should redesign your chemical, but outline some of the key attributes that one should consider when designing a new one that hopefully will be safer. Now, if we're developing molecular design guidelines for safer chemicals, and that's sort of our goal, we can ask, the first question we would ask is, what's in our toolbox that we can already use towards this goal? In other words, we don't want to reinvent the wheel. So what is it that already works well that we could apply as a strategy? Now, outline a couple of these from this slide. We can look at physical chemical properties, and Marty already mentioned that we can use spectroscopic data, the two, of course, being closely related, one being manifestation of the other, really. We can also use results from in vitro and in chemical assays of chemical reactivity, or we can model molecular interactions that we stipulate would be occurring in the living organism and responsible for the toxic response. Now, if we look closely at these four approaches that I outlined on the slide, we can see that computations in one form or another are present in three of them. So they are very important. The determination of physical chemical properties, molecular attributes, modeling of chemical reactivity are the two most obvious ones, but even if we build models based on spectroscopic data, then the quantification of these models will probably involve some form of computations. So if computations are that important, let's look at them in more detail. Now, here I would say that we are very familiar with the paradigm of arriving at a solution to a particular problem by an experiment. Now, here we present an alternative hypothesis to those of you who are not familiar with computations and computational theory. We can solve any problem by computation, and computation being an application of theory that underlines a particular phenomenon we are attempting to understand. So what is important to realize is that computations stem from the theory, while experiments are typically used to define it. So there will be no computations without theory, and there will be no theory without experiment. And it is important that the interconnection and dependence of experiment theory and computation is validated for any problem we attempt to solve. In other words, before applying a particular level of theory to an experimentally unknown solution via computations, it is essential that we apply the same theory to situations where experimental information is in fact available. So that's sort of like a philosophical spiel behind how we can link these three counterparts together and move on to building our model, which is going to be my next step. So at the molecular level, the events that underlie biological activity and toxicity of a chemical are indeed chemical events. So when discussing computations in this presentation, I think it's the most appropriate to talk about computational chemistry. Now, the central dogma of computational chemistry is to understand chemical structure its dynamics, and its reactivity. So an example, say we are interested in the chemical in the left upper corner, the benzopyrene diepoxide, which is known to be mutagenic in humans. We can begin by defining its equilibrium geometry. Next, we may want to investigate its dynamic properties. In other words, how it behaves in certain environment under certain conditions. So, for example, say we would model a specific temperature in the human body that's 37 degrees centigrade. We would model its environment, so that would be solution of a particular composition. We are mostly water, so say we would model an aqueous solution, and so on. And lastly, and I would say most importantly, we want to know how the energetics or the likelihood of the interaction of this chemical with the various elements of its environment, which is often the factor that's responsible for the trigger that leads to the toxic response. So in the case of benzopyrene, we know that that might be an attack on DNA, and the reactivity with nucleotide bases may be the trigger that we are looking for. So to carry out all these computations, we then need a theory that would define the chemical structure, its electronic population, and help us understand the dynamics and the reactivity. So at the beginning of my talk, I mentioned that to design safer chemicals, we might often rely on broad sort of approaches because we are just trying to, capture many different modes of action, mechanisms, or species under one rule. So as an example of such approaches, I want to discuss how one might go about establishing basic design guidelines for reduced aquatic toxicity. I think that's a good example that's relatively straightforward and a good demonstration for this talk, given the time constraints. So this approach is based on defining property-based filters that delineate safe chemicals from toxic ones. What do we mean by property-based filters? So if you take any chemical and you look at its structure, you can characterize that structure by a myriad of physical chemical properties. Most of you will know a lot of them, like molecular weight, volume, surface area, but there are also various electronic properties or parameters. And again, this is something that Marty mentioned before, that would depend on the distribution of electron density in the molecule, such as dipole, or you can look at orbital populations and so on. And on the previous slide, I mentioned that dynamics and reactivity play an important role in the biological activity a chemical may have. Now, what's great about properties is that they can tell us a whole lot about both dynamics and reactivity. So for the purposes of the demonstration here, of how we can use property-based approach successfully in developing design guidelines for reduced toxicity, let's keep it as simple as possible, and let's focus on two properties that we could use. So one of them is log P or log D, which is the partition or distribution coefficient between octanol water. It's a frequently used measure of lipophilicity, which in turn informs bioavailability, so how easy it is for a molecule to pass some membranes. And the difference between the two being simply that log D takes into account ionized species of a particular structure. So depending on your pH, you may want to go with log D over log P, but the principle is the same. Now the second property is the energy of the frontier orbital. So the highest occupied molecular orbital, or HOMO, and the lowest unoccupied molecular orbital, or LUMO. Now most biological reactions, as Marty mentioned, can be defined as reactions between a nucleophile and electrophile. And xenobiotics can act both as nucleophiles and electrophiles, although you'll probably find more reactions where they are electrophiles. And in these processes, the HOMO and LUMO orbitals often take part in driving the chemical reactivity, where the HOMO would interact with the LUMO orbital to form the covalent bond. So the new molecular orbital with lower energy than the original HOMO-LUMO pair. This is basic Gen Chem 101. So the more favorable the orbital overlap, the stronger the interaction. Now, since the Homolumo gap is defined by the two orbital energies, there are two ways you can shrink the gap and make the reaction more probable. That's raise HOMO or lower LUMO. So for any toxicant low LUMO orbital and higher HOMO energies promote reactivity. In other words, the smaller the gap between the HOMO and the LUMO orbitals, the more reactive the compound is. And that's sort of the bottom line of this slide. So now that we have chosen our approach for developing design guidelines, let's choose a suitable system. Now, for acute aquatic toxicity, which is a great endpoint, frequently used to estimate, for example, hazard related to chemicals in surface waters, we have to look at well, where we can get the data that we would use to build and train this model. Now, I have to say that before going further, that this endpoint is a very complex one and involves many mechanisms or modes of action, MOAs. So in some ways, it's very applicable to what we are trying to do here, which is to create a broadsword property-based approach. Now, thanks to the US EPA, we have a lot of data that we can use. We have chosen about 555 chemicals for which we have LC50 values. And these chemicals are broadly categorized into 4 categories of concern based on their toxicity. Red meaning the most toxic, green meaning safe. So now that we have chosen our set, let's see how we can proceed next. Now, before building a model, one might be always tempted to reach for the low-hanging fruit. After all, you don't want to reinvent the wheel or get yourself stuck in something too complex if you don't have to. Now, the most obvious low-hanging fruit would be a relationship between a specific mode of action, or MOA, and toxicity. Now, as this figure clearly shows, there really isn't any really in our data set. For each of the 8 types of MOAs that are represented in a data set, the distribution of LC50 values is relatively broad. Again, on the y-axis, the LC50 is a log, so the bars showing you the spread under each category are actually quite large. So let's move on with our model. So if the MOAs or the modes of action didn't provide any useful information, let's look at some of the properties which we know contribute to toxicity. So let's look at log P and log D. And if we do that, we start seeing much more meaningful trends. So that is that chemicals that are more toxic have higher log P, log D values. In other words, they are more lipophilic, and so they can more easily pass through cell membranes. Again, as I mentioned before, log D reflects the contribution of ionic species, and since roughly 15% of our data set is ionized at pH 7.4, which is the biological pH, it makes sense to continue with log D rather than log P. Now, the important point to make here is that as much as we are starting to see meaningful trends with log P and log D, the overall overlap between the EPA's categories of concern, in other words, the colored boxes that you see in these 2 box plots, are still far too great. Even when we consider category 1 highly toxic red and category 4 safe, we cannot really distinguish between high probability, we cannot distinguish with high probability which is which based solely on these descriptors. And again, this is something that I think makes perfect sense, since bioavailability, which log P and log D estimate, is only one of the stipulations for observed toxicity. So we have to go on. This is a good start, first step, but let's go on with our property-based filters. So let's throw in our second property-based filter, which is the Homolumo Gap, or the gap between the 2 frontier orbitals, and which we stipulate represents chemical reactivity. Now, if we do that, we get these 2 box plots that you see on the slide. What is interesting is that the safe chemicals, which are labeled in green again, are pretty much all confined to the left upper quadrants of these plots. Again, as for rationalizing this observation, it actually makes a lot of sense that you would find safe chemicals in this region. These are the chemicals that are more water-soluble or hydrophilic and have a large homolumo gap, so suggesting that they are less reactive. Now, if we carry out a bit of statistical juggling, we can define this region as having boundaries of log D or log P less than 1.7 and homolumo gap greater than 6 electron volts. I should say that we could just look at these figures and arrive pretty much qualitatively at the same conclusion. So what does this region mean? And I would say we can postulate that this is something like a safer chemical space, so a region with higher probability of containing mostly safe chemicals. How high this probability actually is, I will show you on the next slide. But before we get there, I think it's good to look at some practical examples, some high-profile chemicals that we know are toxic, and see how they do in our crude analysis. So this is in the table in the right bottom corner. You look at BPA, the poster child for endocrine disruption. It's out of the safer chemical space, so it's predicted to be a toxic chemical on the basis of being too reactive, and so are the other well-known endocrine disruptors such as phthalates, atrazine, PBDE, which was previously mentioned as a toxic flame retardant, which is also interestingly ruled out on the basis of being too lipophilic. So let's look at if we can further improve and quantify the performance of these design guidelines. Now, looking at the safer chemical space, in just the left upper quadrant, we see that the most of the green dots are confined to this region. There are still a few red and orange dots, in other words, chemicals of moderate to high concern for aquatic toxicity. So say we want to search for a third property that may eliminate these chemicals from the safe region, we will find that molecular volume is actually a pretty good candidate. And that's what's shown in the box plot in the middle. You can see that it eliminates the high concern chemicals from the rest, and even the moderates are pretty well separated from the safe in green color. Now, this time it is harder to rationalize the importance of this property filter to see why chemicals would be more toxic if they get larger. But I would say that in this case, the volume really is somewhat of a coincidental placeholder for something else, something that is not just very well described by the model, which typically means metabolism. Now, if we accept the stipulation that we have a new three-property-based model, which we can not only separate safe chemicals from the rest better, but we are starting to see separation between categories of concern in general, which is best shown in the graph in the upper right corner of the slide. Now, if you focus on the table at the bottom, then it sort of summarizes the performance of the model as we went through all the steps to develop it. So in the first row, you can see how the chemicals are distributed in the data set without any filters being applied. In the second row, we can see that when we apply the criterion of the log D being less than 1.7, we marginally reduce the number of high concern chemicals, but significantly reduce chemicals in the moderate concern category, while still retaining low and non-concern chemicals at the same time. Now, if we apply to that, on top of that, the Homolumo Gap criterion of greater than 6 electron volts in the third row, we cut the numbers of high, moderate, and low concern chemicals more or less by half, while we still retain an impressive, I would say, almost 90% of no concern chemicals. And the last property filter eliminates all but really one high concern chemical in the data set, while marginally reducing moderate and low concern categories as well. So overall, roughly 88% of safe chemicals are retained, which indicates that even as simple as they are, these design guidelines have a high efficacy in terms of false positive rate. And with regards to false negatives, the only high concern compound in the data set that remains in the safer space is an allylic alcohol. It's the 1%, which is postulated to undergo metabolic conversion to acrolein in liver. And acrolein is known to be much more reactive, and it's actually ruled out by the Homolumo Gap criterion. So this is a very good start. Another way to look at the performance is to look at the last two columns, which show the mean LC50 value in the safer chemical space. You can see as we apply these filters, the values go up significantly, which means the compounds contained in that region are less and less toxic. So in summary, how good are these design guidelines? So if we are to summarize this, the compounds that meet the property-based criteria are about 10 times more likely to have no or low acute aquatic toxicity compared to the compounds that do not meet these criteria, in other words, compounds that fall out of this safer chemical space that we defined with the three properties. There is less than 1% chance that chemicals belonging to the high concern category for aquatic toxicity are included in the safer chemical space. So this is sort of a couple of interesting box plots. So I've mentioned at the beginning that there are many modes of action involved in this data set, in this endpoint. So if we look briefly at how the property-based filters do for the four of the most representative ones. So for narcotics, the boundary of the log D eliminates nearly all of the high and moderate concern chemicals, while the remainder is excluded by the Homolumo Gap criterion. A similar pattern we observe in the case of polar narcotics. For electrophiles and proelectrophiles, the log D screens out 21 out of the 23 high and 30 out of the 61 moderate concern chemicals, adding the Homolumo Gap criterion as an additional filter screens out 1 of the 2 high, 12 of the remaining 30 moderate, and 2 of the 8 low concern chemicals, retaining the single no concern chemical. Now, this is all, a lot of numbers, but the most important one to remember, or the most important one to focus on, is actually the last box plot, which is the MOA not determined. So in the case of the MOA class that was not determined, the volume criterion actually played a significant role. So again, suggesting that volume may be a placeholder for something like a metabolic activation. Now, as with any tool, it's always important that we validate it on a different set of data to see how robust its predictive power is. And if this approach of property-based filters is indeed a well-functioning broad sort that applies to a wide range of chemicals, then perhaps it should work on an organism at a different trophic level. So the one we chose for the quick validation here is Daphnia magna, which is a species that's also often used for aquatic toxicity endpoint. And the data comes from the Japan Ministry of Environment. We actually looked at 363 chemicals, which compared to our training set of over 500 chemicals is a pretty substantial validation. Now, what's important is that only about 15% of these chemicals overlap with the chemicals that we use to define this model. So, although not truly external validation in a Puritan sense, it's very close to it. And the results are compared in the table that you see there. So, for both the general observations are really the same. So we are consistently showing that 1, nearly all the high toxicity category compounds are excluded, and 2, increasingly more compounds are eliminating, proceeding from none to high toxicity category with the application of our property-based filters. And somewhat lower the ability to retain the compounds in the none category. If you look at the last column, the 67% for Daphnia and 88% for what is fathead minnow. So that's our trainings that you can attribute that to having very few compounds of this category in the validation data set overall. So, this was a sort of a very simple demonstration of how we can proceed, certainly not the answer, but how we can proceed towards developing design guidelines for safer chemicals, or sort of a few rules that we could apply at the design stage of a new chemical to make sure that we avoid the rotten apples. And again, these would be very different if applied to different toxic endpoint, but it's the thinking, I think, that could translate from one endpoint to the other. So 3 take-home messages. I think we can, sort of show that we can build simple guidelines for reduced toxicity that can be applied to the design of new chemicals. 2, we do not need a multitude of descriptors, as commonly seen in many QSAR models, to obtain valuable probabilistic information regarding chemical toxicity. And 3, which is my favorite, is that the simplicity of these guidelines means that this provides a lot of benefit to chemists that are trying to retain functionality in their molecules, but while designing around toxicity. In other words, all of these 3 properties I mentioned, they can very clearly relate to the structure they are dealing with, and modify accordingly to end up with one that's safer. So again, not the answer, but a start towards hopefully approaches that are proactive and more successful than assessment of alternatives. So.
Excellent, thank you, Jakub. Our final presenter is Nigel Greene, who will be talking about incorporating safety into early drug design. Nigel?
Thank you. So I guess in the interest of time, I'm going to move relatively quickly through some of the slides and hopefully make sure I've got control. Yep. So basically, coming from a pharmaceutical industry perspective, the attrition in our R&D process is actually pretty high. So we can take sort of millions of compounds if you look across the discovery approaches that we have within Pfizer, and basically come out with about 1 or 2 products out of those millions of compounds that we do that. And it obviously takes a long time to get there, and so selecting the right compound and designing the right compound is critical to the survival of the industry moving forward, not just for Pfizer, but for everybody, to get novel medicines out. So if you think about the costs involved in developing a new medicine, and I think nowadays the current average cost of new medicines is about $1.3 billion per drug, which is an enormous amount of money to spend, particularly when you think about the time involved and the survival rate of novel compounds. So it takes about 10 to 15 years to get to market, by which time you probably have about 10 to 15 years worth of patent life left in the molecule, and at which point only 1 in 5,000 to 10,000 compounds is actually going to be successful in being approved. So the success rate is pretty low, and so it's absolutely critical to consider not just the pharmacological action of the drug and how it treats diseases, but also the adverse events that might be associated with that. And so what we've instituted within Pfizer is sort of in parallel to our drug discovery process. And if you think about the typical sequence of events that go on, all the way from selection of an appropriate target or protein to go after, you've got the screen development and high-throughput screening to identify potential hits. From there, you'll sort of narrow that down and do, I guess, hit validation to get to what you feel are significant lead series. And then move into lead optimization and eventually select your compound to push forward into preclinical and clinical trials. At which point, what we're trying to do here is assess the safety of those compounds in parallel to optimizing both the bioavailability and the potential efficacy of that compound. And so we'll conduct target assessments to make sure that the pharmacology itself for the primary indication is going to be safe. We'll use in silico and in vitro assessments in the high throughput processes to sort of try and optimize the chemistry both for safety, and obviously we'll do some early in vivo toxicity testing to try and characterize that. And similarly, as you encounter safety issues moving through that pipeline, you might go back and sort of develop new assays to help characterize the potential risks or safety issues that you might have. Just to reinforce sort of why safety is so important, if you look at the key causes of drug attrition across the industry, and things haven't really changed in the last 20 years, safety is still one of the primary causes of compounds being removed. And in actual fact, 10% of new chemical drugs show serious adverse drug reactions after market launch. And so even once you get it through the clinical trial process, you haven't necessarily eliminated the risk of failure. And that's a very expensive experiment to conduct, which is, essentially you spent $1.3 billion to reach a drug that is then withdrawn pretty early for safety issues. So the basic question that we ask is, what are the design features which can signpost risks for safety in the clinic? And that's actually quite a challenging question to ask. And we've been thinking long and hard in the industry, and Pfizer's no exception here, about what they might be. And essentially it narrows down to, there are actually 5 key elements. The 5th one is metabolism here, but you can argue that the metabolite of a compound also experiences these 4, which is, what is the primary pharmacology of the compound, i.e. the intended target and the safety of that intended target. What are the secondary pharmacology events of that compound? So what other things is that compound interacting with in terms of proteins and receptors? You could call it that off-target or unintended pharmacology. All of those titles are perfectly valid. The 3rd one is chemical structure. So there are sort of inherent liabilities with chemical structures in terms of reactive metabolites, and things like that can cause protein adducts, stimulate the immune response, and cause sort of adverse events through an immune-mediated mechanism. And then obviously there's the physicochemical properties, which we've heard quite a lot about, where they can also cause or introduce liabilities that you don't want in a molecule, such as phospholipidosis or QT interval prolongation, which can lead to cardiac arrhythmias, which certainly don't want patients dropping dead rather suddenly on you if you can avoid it. So in terms of structural alerts, if you look at the 81 drugs withdrawn for idiosyncratic, or what is termed idiosyncratic toxicity reasons, well over half, 67% of those all contain structural alerts, and only 5% have no reactive functional group. And so introducing some of these key groups which lead to sort of reactive metabolites is definitely a problem, and probably should be avoided wherever you possibly can. Obviously, sometimes that's not always possible. Sometimes reactivity is intended in a molecule because of its use case. But if you can avoid using potentially reactive functional groups, then your risks subside pretty dramatically. In terms of physicochemical properties, some of the things that we've noticed here, and again, there are numerous publications in this area looking at log-P and topological polar surface area. So some of the rules that we've come up with is that if you have, I guess, topological surface area of less than 75 Armstrong squared and a C log P of greater than 3, then you're 6 times more likely to see toxicity in vivo at low exposures at a Cmax concentration. So that's a plasma exposure of less than 10 micromolar, which is pretty low. Similarly, if you're thinking about off-target promiscuity, this is also linked in some part to the physicochemical properties. And in this case, you have polar surface area and C log P. Again, if you are in that bad space, that upper left quadrant, then you are 25 times more likely to hit more than one pharmacological target in binding assays, and that's a greater than 50% inhibition at 10 micromolar when you look across the compounds. And so the odds are pretty high, are stacking against you as soon as you start introducing lipophilicity into a molecule or low polar surface area. In terms of being able to characterize promiscuity, we've done quite a bit of work now looking at what's sort of the optimal sort of size of screening panels. Obviously, you can't test, or it's financially not viable to test against every single possible protein that you can think of to understand whether you're likely to interact with it. But in essence, you can come up with small panels of, say, 15 or so targets, which would give you a good indication of your likelihood of being promiscuous across a larger range of targets. And so here on the x-axis is the promiscuity index based on 15 targets, and on the y-axis is the sort of the full profile, which is about 184 targets that we look at in the Serot bioprint profiles, and you can see there that the correlation is pretty good, that if you're promiscuous in the 15 targets, you're more than likely to be promiscuous in the 184 targets. And so there's a pretty good stands to reason that using just 15 targets, you can get a good sense relatively quickly of how promiscuous your compound is going to be. So this is where I now get on a little bit of a soapbox that I have, which is when you talk about in vivo data and thinking about dose versus exposure, when you actually look at some of the outcomes from in vivo experiments and look at the dose versus the Cmax, so that's the plasma exposure, when you look at compounds that were dosed at 50 mg per kg, you can get up to 5 orders of magnitude difference in terms of plasma exposure. And so the bioavailability and the absorption and the clearance and the distribution of the compound all play a critical role in whether that compound is going to be toxic or not at that 50 mg per kg dose. And so just looking at doses within in vivo experiments doesn't really tell you anything about how much exposure in the bloodstream there is for that particular animal. There was a very nice paper from some folks at Lilly recently in JMed Chem, which was looking at the lowest observable adverse effect level in rodents and asking what properties drove that determination to be much lower in the in vivo experiments. And what they found through their analysis was looking at pairs of compounds that from the same chemical series, so you negate the effects of pharmacology in there, that volume of distribution, so how much compound is getting into tissues, actually the higher that the volume of distribution, so the more compound that gets into tissues, the lower the LOAEL is going to be. Similarly, if they saw an increase in LC50 in a cytotoxicity assay, then you saw an equal increase in the LOAEL. So cytotoxicity also plays a key role in determining where your lowest observable adverse effect level is going to be in a rodent study. And so these are two things which are both measurable and calculable from physicochemical properties and chemical structures. However, one of the things that you have problems with in these in vivo experiments is you're very much at the mercy of which doses were set as part of the study design. And typically, most in vivo studies that we run here in pharmaceutical development have typically three doses: a low dose, a mid dose, and a high dose. And this plot is showing sort of the relationship between dose and the Cmax. And as you can see already, that the Cmax value is not linear to dose. So you can increase the dose and eventually you plateau out and you don't get any more compound getting into the bloodstream. And so that inevitably your exposure plateaus off at the higher concentrations, further confounding the issues around what the actual exposure was observed for that. But if you look at the two points that are marked there, you could argue that if that mid dose was the LOAEL at a 30 mg per kg dose, and the 3 mg per kg dose was an NOAEL, then you would sort of argue that the exposure for your low L, you could take that value. But if theoretically, if there was a concentration where the toxicity started to occur, sort of shown by the red line, then you could argue that your theoretical no-L dose could be much higher than 3 mg per kg. And so arguably, sort of the rate of severity of the toxicity within the study may or may not be linear between those doses, but where it starts to become significant can play a role in where you set your no-L and low-L dose. And so depending on how you set those doses, you may get very, very different answers for different compounds. So what we've tried to do here at Pfizer is come up with a system for looking at all of the observations made in vivo studies and assigning them a sort of an arbitrary scoring system where, based on the severity of or the impact of each finding, so things like a redness of skin would get a very low sort of impact, sort of score of maybe of 1, whereas if you saw actual inflammation, it might be a score of 10. Degeneration of tissues would be 100, and say death would be on a sort of an order of magnitude bigger of 1,000, which is really about as bad as you can get. And so you could argue, and going through this process, you start to score the findings that you see at each individual dose group, and you come up with a score that you can then start to plot in a dose-response relationship. And using the scoring system we came up with, we set a threshold of about 100, which is where you start to see what we would call significant levels of toxicity, so pathological damage typically, or multiple clinical chemistry parameters going sort of out of the normal range. And at this level, you can then sort of draw, if you plot both the dose and the severity against each other, you can draw a threshold, and you can ask, where does that dose-response curve cross that threshold, and you come up with this concept of a toxic Cmax, which is what we do. There is a slide in the backup to talk about why we use Cmax rather than AUC, and to be perfectly honest, it doesn't really matter. Both values correlate with each other. AUC is a continuous measure over a period of time, and so you can, or you can use C average, but in essence, they all correlate very well together anyway over the range of concentrations that we're talking about, and so using one versus the other doesn't really make a difference. So this is sort of the toxic Cmax approach as a graphical concept. So we come up with a severity score for each dose. We've plotted against the concentration on the x-axis and come up with sort of where sort of the highest point that falls below the threshold and the lowest point above the threshold. We'll draw, rather than doing a curve fit based on 3 points, which is, I guess, mathematically challenging, and subject to error, we just draw a straight line and we get pretty much the same answer anyway. Drop down onto the concentration axis and you get what we call a toxic Cmax value for a particular compound. And now this allows us to put all of our studies, or the compounds that we've studied in vivo experiments, on an equal footing. So we can look at this value for every compound in every species, in every duration, and every study that we've run to look at how does that change and what properties tend to correlate with these values. So the first question we ask is, well, how much does this value really vary when you run multiple studies on the same compound? And in actual fact, it's relatively good, which was surprising to me actually, that most of the values where you've got repeat studies are they're within a threefold difference, which is probably within biological variability of the study anyway, bearing in mind that you're talking about a very complex experiment to run. And so we were fairly confident that we could get within about a threefold. Obviously, when you're doing modeling work, you're not going to get within this sort of variability, but at least we sort of have defined sort of a range where we can get close to. So we started looking at properties that would correlate to this toxic Cmax value, and here the red lines and red dots correspond to a toxic Cmax, compounds with toxic Cmax values of less than 3 micromolar. The yellow is between 3 and 30, and the green is greater than 30. And so here you can see, again, on the left-hand side, the volume of distribution. The higher the volume of distribution, the more likely you are to be toxic at lower concentrations. And probably arbitrarily, you could draw a line at around 2 and say that once you get above a volume of distribution of 2, things start to go a little bit hairy and your toxicity comes down to very low plasma concentrations. Similarly, this seems to correspond very much to promiscuity. So if you plot volume of distribution versus promiscuity in the right-hand plot, you can see there that the compounds that are very promiscuous, as sort of more towards the lower end, sort of I guess the lower part of the y-axis, so the lower values of this Gini coefficient correspond to greater promiscuity, higher values correspond to lower promiscuity. You can see there that volume of distribution and promiscuity are inherently linked together, that your volume of distribution, it's basically measuring stickiness to proteins and your ability to interact with multiple different proteins within a biological system. We then looked at things like cytotoxicity and how well different cell lines sort of pick up on compounds which cause toxicity. And so here, again, the x-axis in all 3 plots is the LC50 in different cell lines. The cell lines are the headings above the plots, and basically the curves are is a density plot with the number of compounds. Obviously, the higher on the y-axis you go, the more compounds fall under that part of the curve. And so here you can see that the HepG2 cell line looks pretty good. So if you're relatively cytotoxic, so less than 100 micromolar LC50 in HepG2, you're going to start to see toxicity at a plasma concentration of around 3 micromolar. Similarly, you see the same sort of thing for NRKs cells, but for THLE cells, it's not quite so good. It's not quite differentiating as well. However, this is on sort of the global world of chemistry, and when you start to split these things out, as you'll see on the next slide, that you see very distinct differences in what those cell lines are picking up on. So here we've actually separated the data set into acidic and basic compounds, and now you can start to see that HepG2s actually pick up the toxic basic compounds pretty well, that again less than 100 micromolar IC50, and you start to run into sort of issues with compounds being predominantly toxic at concentrations less than 3 micromolar, whereas THLE cells actually do pretty well with the acidic molecules. So they do differentiate much better than HepG2s for the acidic molecules, which are sort of less than 3 micromolar in terms of their toxic Cmax values. So we then sort of decided to use random forest modeling to just ask the question, so what variables really dictate this or are important in coming up with this thing. And so there's a whole number of different variables, but in essence, the key properties that came out of the mixture was actually it was a calculated volume of distribution at steady state. The pKa value obviously plays a key role, and the polar surface area divided by molecular weight, which is really just talking about sort of a 3D polar surface area, if you want, but it's far less computationally intensive to get there. And so it's easier and faster to just take the topological polar surface area and divide it by the molecular weight, and you pretty much get to sort of an approximation of 3D polar surface area. And so you can start to see where we're heading with this, and we actually went ahead and developed a model, and although not great, actually does put compounds in the more or less in the right ballpark for a lot of things. There are some compounds obviously where we're not predicting very well, and we suspect these are usually because of toxicity caused by the primary pharmacology or by very potent off-target pharmacology events, or could be due to active transport into tissue. So the tissue level exposure is much greater than you would see in the plasma, and so you would end up, due to active transport, some of these are actively transported by OATPs into liver, for instance, and so you're going to have a greater concentration of liver toxicity or compound in the liver, leading to toxicity at lower concentrations in the plasma. But here you can see, on the whole, the blue lines are a threefold sort of, I guess, the threefold difference, which is about the experimental variability in the values themselves, and the red line is a fivefold, and so the majority of compounds were at least getting within fivefold of their actual values, which at least puts us in the right ballpark when you're talking about where you're likely to see toxicity occurring. However, in drug development, most decisions are not based on sort of an exposure of where you see toxicity. It's based on what we call a therapeutic index or a margin of safety or whatever you want to call it. But it's in essence the difference between where we would expect to dose patients for efficacy, for treatment of disease, versus the concentration at which we think we're going to see toxicity, and that the larger the TI, the bigger margin of safety you've got, and the more likely you are to be okay moving forward into the clinic. And in general, our rule of thumb is about a 30-fold TI for most events, with some exceptions where some, if they're not monitorable in the clinic, they will be much more cautious about their events, and obviously if they're sort of very sort of severe effects, will also be very wary of taking those compounds forward without an adequate TI. And obviously the actual TI level that's suitable for a particular effect has a lot of different factors that will influence that. So the indication of the drug will play a role. So if it's a life-threatening versus curing a common cold, the duration of the treatment also makes a difference. So if you're going to take the drug for the rest of your life versus a drug that you take for 7 to 10 days, the TI level would be critical. And similarly, the patient population. So if you're talking about elderly patients that are on multiple therapies, then obviously you need to think about some of the issues there. However, in doing the modeling effect, when you look at our ability to predict which compounds would be successful based on the TI, we actually do a pretty good job. So the top two pie charts here are the actual TIs from the experiments based on the observed toxicity in vivo versus their projected human efficacious concentration. And so obviously just an arbitrary line of 30-fold to cause a cutoff, but you can see here we get more or less the same numbers in both sides of that 30-fold. There are some slight differences that some compounds we predict safe, they ended up with a less than 30-fold, etc. But in general, we would have not thrown away too many good compounds by using a in silico approach like this, or a combination of in silico and in vitro approaches to develop this. So just as a sort of the penultimate slide, I do want to come back to the fact that in vivo toxicity is multifactorial, or mostly multifactorial. There are some instances where it is a discrete mechanism. If you think about triglycerin, which is probably the most studied drug in the literature right now, which was withdrawn for liver failure, I guess back in 2000, this compound has a lot of things that are wrong with it, and all of them have probably led to the liver failure. I should point out that we didn't know a lot of these effects up front. Through the experimentation and the research that this compound triggered, have led us to understand more about what causes sort of liver failure in humans. But this compound will inhibit the BSEP transporter, which is the bile salt extraction pump, which removes bile acids from your liver cells into the bile duct and out through the gallbladder. The physicochemical properties of this compound are pretty awful. It's very lipophilic and not particularly nice to deal with. It also inhibits mitochondrial function, so it's both an uncoupler and an inhibitor of mitochondrial dysfunction, which can lead to reactive oxygen species and glutathione depletion and cellular stress. There's a reactive metabolite formed through quinone formation at the tail end of the molecule there, which also leads to glutathione depletion and cellular stress and cell death, and also to covalent adducts to cellular proteins, which then form, haptens, which can trigger an immune-mediated response leading to liver failure. And so it can be very complicated to come up with what the mechanism of toxicity actually is, but similarly, a lot of these features and properties are things that we now know about, and we should probably sort of avoid using some of the known issues that we have. So just to finish off and summarize, we've been using this approach now across our portfolio, and we believe that we can really predict the 60% of the compounds which cause low-dose toxicity in preclinical species with a fairly low false positive rate. And so this enables us to focus our resources on those compounds which are most likely to succeed. We've been using this since probably 2010 across more than 70 discovery projects. But what it's really doing is initiating safety considerations early in the project. So people are starting to think about the safety concerns much earlier. And then obviously building this tool actually relies very heavily on having a well-characterized training set of compounds that we can use. And so it does require a lot of heavy lifting in going into sort of our databases and pulling those out. And in essence, the value here is just really steering away from the chemistry that's never going to be successful. And so we can guide our medicinal chemists to series or chemical space, which is going to be much better for them to use. And with that, I'm going to finish.
Great, thank you, Nigel. We have a question, and one of them is, how do we more effectively take this information that we're learning about function, structure, and toxicity and integrate it into design? What gaps, information tools, and training are needed?
I'll take a first shot at that. This is Marty. So a lot of the answer to that question also depends on what sort of team that you're working with at your individual company and what sort of products you're actually developing. So the tools and the information that are coming out of this molecular level toxicity and understanding computationally what the most important factors are that affect potential toxicity of a molecule can really then be translated into filling in some of the data gaps that exist in current hazard characterization for the problems that a particular company is facing. So I think that, you know, the first thing that's needed to use this sort of information at the molecular level is a clear definition of both the end state that you're trying to get to and also what information is and is not currently available. So this stuff is really good at helping you bridge the gaps in current understanding about toxicity. It helps pharmaceutical people decide what the best types of molecules are to develop. It can also help you decide what the best among a bunch of options are in order to use or choose.
Jakub or Nigel, do you have any additional comments?
So, I guess I'd agree there around the fact that it does come down to what the use case is and the problem that you're trying to solve as to the approaches that you can develop. I think thinking ahead of time about the physicochemical properties of a compound is really, really critical. Depending on what's tolerated, the physicochemical properties really do dictate the amount of absorption and distribution, the rate of clearance, for instance. You've got to, those are the key elements of design. Small changes, lowering lipophilicity can do the world of good if you're looking for things that are less toxic at low plasma concentrations. Similarly, solubility is going to be one of the issues there as well. So, thinking ahead, depending on what your use case is, you've got to think ahead about what's the impact of the physicochemical properties going to be on my compound.
Great, Jakub, you?
I would agree with what was said before. I would just say you have to find a common ground between your function. You have to find a common ground between the function and the toxicity, and figure out what it is that's driving your function. And quite often it's a few simple properties, and if you can find a common ground where those properties can still be retained while you move yourself to say a safer chemical space, you move yourself out of potentially creating a hazardous chemical based on the approaches that already exist that have been published. A couple of the basic rules. I think that's a good place to start, and always finding a friend who's a computational chemist who can produce some of those descriptors for you, calculate some of those properties if you can't measure them. That's something that could be always helpful.
Thank you. And one last question is, Jakub, is your technique still under development, or has it been used today to predict toxicity of new chemicals?
So this one actually is already used. It's incorporated into a tool we call Toxaqua, and what I presented today is simply a basic framework for it, but the tool itself addresses much more than just the safer chemical space. It can also distinguish the individual categories of concern, so we can predict with about 85 to 90% probability whether the compound's going to be moderately toxic, high toxic, or low toxicity. So it's in science, the wheels never stop turning. So there's always progress moving forward. But as it is, it's a developed tool.
Okay, thank you. Thank you again to our speakers, Marty, Jakub, and Nigel, for your wonderful presentations and for your comments. And thank you, everyone, for attending.